Everybody, this morning we're going to be doing in the Barcelona Supercomputing Winter School a section on molecular medicine. This is part of the Comp Biomed training, and my name's Andrea Townsend Nicholson, and I'd like to introduce you to Art Hoti, who is here with me, and we are both from University College London. We're both in the Division of Biosciences, Structural and Molecular Biology. So yesterday, as Carlos was telling you, you were doing the computational part of computational biomedicine. And now we're going to move you more towards the biomedicine part of computational biomedicine. So this is a brief introduction to help you understand one particular example that we're going to be looking at today in a hands-on session. And before we get started, you will need to download some data for this. You'll need to transfer it from the projects directory across to your own account. So what I'd like everybody to do before we get going, and then give me a hands up, please raise your hand when you've done this, is to, at this point, log into your Mare Nostrum account, go to your home directory, and then copy the files that you need for this workshop across to you. And this is exactly the same format that Carlos gave you for copying Okba's labs folders yesterday. So you just do this here, except that you're in a different account. We are working from NCT 0005, and we are going to be looking at cheese. So if people could please copy this across, then we can get started, because it does take, there are some large files in here and it will take a while to move them. Is everybody comfortable with the copying process? Could you please put in the chat if you're not comfortable with the copying process? Okay, so I assume that everybody is able to do this, and we will then carry on with the presentation. So to give you an understanding of why somebody would be working at an interface between computational and biomedical or biomolecular science, I thought I would give you some context about what I'm interested in personally, and my research is aimed at providing opportunities for personalized medicine. And I work at the molecular scale, so I'm interested in understanding how small molecule drugs interact with receptor proteins to execute particular biological functions. And you will hear, after we have our hands-on workshop, from Alexander Heffetz of Evotec, who is working in a drug discovery company and will be providing with you with some more information on how to be able to use computational methodologies to design drugs. So I'm not just interested in doing it from the research side, I'm interested also in doing it from the training side, which is why Art and I are here with you today. So to be able to start off, I wanted to explain to you what the overarching vision is that the Comp Biomed Center of Excellence, so we're a Horizon 2020 funded Center of Excellence in Computational Biomedicine, and I wanted to help you understand what our vision is and why we are so interested in working on computational biomedicine. So I'm going to stop sharing this screen now and show you a film that encapsulates what you're doing to help, what we're doing to help you understand better. So we need a film, which we will get to. Can people see this in full screen?
No, it's in presenters mode.
Okay. For some reason, Chrome doesn't seem to like to do this in full screen. Right, okay, well, here we go. You can see it now as the inset in YouTube.
It's still the presenters mode.
Oh, is it? Okay, that's interesting. Let me sort that out. How about, can we see a virtual human YouTube now?
No, now it's the PowerPoint slides.
Okay. Oh, how interesting. Sorry about this technical fun and games.
Okay, now it's YouTube.
Excellent. Thank you. Off we go.
Our bodies are exquisitely complex machines. In our efforts to care and improve them, we look to those who push the limits of what is humanly possible, such as athletes. Now, you would say, what can we learn from them if every athlete is different? If they achieve peak performance through customized diets, training, and technology. As it turns out, we too are all different. That is to say, there is no single lifestyle, no diet, no medication that will work equally well for all of us. If this is so, how can we get the best out of our bodies? Now, imagine a virtual human, not made of flesh and bone, one made of bits and bytes. And not just any human, but a virtual version of you, accurate at every scale, from the way your heart beats down to the letters of your DNA code. You know that many drugs only work well on some people and can cause serious side effects in others. The reason is variations in DNA, our genetic differences. But we understand how these DNA differences change the building blocks of your body, the proteins, and we can simulate in a computer how drugs interact with them. By testing drugs on your virtual body, your doctor may eventually be able to test a wide range of drugs and select precisely the right one to suit you. But sometimes, choosing the right drug is not enough. We must also be able to deliver it to a precise target in the body. Take inhaled drugs, for instance, which can end up in the walls of your nose or at the bottom of your lungs, depending on the intricacies of your particular respiratory system. Using supercomputer simulations based on scans of your lungs, we can predict with high precision where particles will flow, and then design devices that can deposit drugs exactly where your body needs them. Well, obviously, mastering drug delivery means controlling the body's main transport network, your circulatory system. By simulating the movement of red blood cells and other cells, we can understand important protective processes, like those that prevent blood loss after an injury. Virtual humans could help doctors to plan risky surgery too. They could be used to work out how to reach an aneurysm deep in the brain that is at risk of rupture, which could cause a stroke. Surgeons could then try out the best treatment or implant to suit the location and shape of that particular aneurysm. They could even double-check that the implant will not cause problems, such as clotting, before they try it out on you. Physics, chemistry, and biology intertwine in your circulatory system to drive the most remarkable pump that evolution has ever created, the heart. To create a virtual heart, processes have to be modeled at multiple scales, from the contraction of muscle to the blood flowing through its chambers, to the movement of charged atoms during a contraction. Virtual hearts already beat within supercomputers, where we can test the effect of different drugs or pacemakers, but also more fundamentally, to understand the way an individual's heart works. Your virtual avatar will not only be built in your image, it will also move like you. We can use it to calculate the forces and the mechanical stresses that are constantly induced on your bones, and predict the risk you will suffer a fracture. Doctors can also use virtual humans to work out how diet, exercise, or drugs can help those with brittle bones. And what if we go even further than treating disease, using virtual humans to prevent illness? Within a supercomputer, many virtual versions of you can explore small changes in your lifestyle and how they affect your health, aging, and quality of life. Virtual humans, helping you to figure out the very best that you can be.
So I thought you would particularly enjoy that because it was rendered on the machine you're using on Mare Nostrum 4 at the Barcelona Supercomputing Center. But the idea behind the virtual human is that you can take data from every scale, from the nucleotides of your DNA all the way up to the way in which your skeleton moves, and you can use this information to be able to make a particular medical solution for an individual. So you can have personalized medicine. So let's go back to the presentation now. Which hopefully you can see here now. So that's the idea and the vision of the virtual human. This is another thing you can do. You will recall from the film that they used gait analysis for how a skeleton walks, and they were combining the movement with the X-ray data with information about bone mineral density so that you could work out where fractures would be. This is also what you can do with your skeletons and these sorts of data. So you can have fun with your virtual humans as well. Let's just move on. So the example that I want to speak to you about today, and what Art and I will help you do as an experimental computational piece of work, is to take a look at metagenomics. You may well have heard a lot about metagenomics in the press and in medical articles and in popular science articles, because we're increasingly learning more about the community of microbes that are found in or on multicellular organisms. So a microbiome is the genetic material within this population, and metagenomics is how we study this genetic material that comes from environmental samples. So this is an example of being able to use advanced experimental technologies to overcome problems that previously existed. So in traditional microbiology, you can see in the top portion that there are a number of different places where you can obtain bacteria from. People would isolate and culture these bacteria to study them. But the problem is that most of these strains or species of bacteria were not able to be cultivated. And so very little of the richness was able to be explored by cultivating things in this way. However, what we can do by purifying things directly is to have a better insight into this microbial dark matter. We don't need to culture the bacteria. We can purify things straight from them, isolate the DNA, and use them in sequencing. So this will give us access to all of the genetic resources within an environment and help us better understand the complexity and the richness and how this is interacting with and informing the environment. So direct isolation, in theory, gives us 100% of all of the microbiome that's present in the area that we're studying. Now, you may have seen these kinds of phylogenetic maps. They show the relatedness between species, or in this case, between phyla. And I've just shown you on the right side the increasing order in which our species are organized. This is called taxonomy. So the highest structure is life, and then underneath it sits domains and kingdoms and phyla. And we're looking at a phyla here, a series of phyla. And this is all the related species. So it makes a tree. The closer things are to each other on the tree, the more related they are to each other. You can see that the lower branch has eukaryotes. So those are a diversity of multicellular organisms, including mammals and animals and ourselves. There is a domain called archaea, which are not as complex as eukaryotes, but are very similar to them in the processes and molecular structures that they have. And then up at the top, we have this huge range of bacteria. And it's the bacteria and the archaea that we look at when we look at microbiomes. So if you just took a look at a human being, and we have on the right here a representative diagram of an individual looked at from the front and from the back, there are many different skin microbiomes, and you can see from the legend the different kinds of bacteria. So we have actinobacteria, Bacteroides, Firmicutes, Proteobacteria, all sorts of different things. And you can see from the pie charts for each of the areas where the human microbiomes have been characterized. This is just a general assembly of information, not one particular individual, but you can see that there's a huge diversity of the number and the variety of the different bacteria that are present in different human microbiomes. So this is something that is of particular interest and medical relevance, and this is why we're taking a look at it. So I'd like now to show you another film which will help you understand these microbiomes. Let me just pull this down here. And because people tend to think of bacteria as being bad. We have lots of antibacterial soaps. We have lots of ways that we try and avoid getting bacterial contaminants on ourselves. But this film will hopefully help explain that it's the balance that's important and that some bacteria are good for us. So we're going to take a look at this now. Can everybody see this YouTube?
Yes.
Excellent, thank you.
The next time you look in the mirror, think about this. In many ways, you're more microbe than human. There are 10 times more cells from microorganisms, bacteria, viruses, fungi, than human cells in and on our bodies. And our genes are outnumbered 100 to 1 by microbial genes. Scientists even have a name for all these microbial genes, the human microbiome. Now, This might make a lot of people rush for the hand sanitizer. But it turns out most of these microorganisms aren't bad, germs that will make us sick. Most are good. And without these good microbes, our bodies don't seem to do as well. We don't seem to be as healthy, and we actually might get sick more often. So one question is, where do our microbiomes come from in the first place? Well, like a lot of things, it starts with our mothers. As the infant passes through the birth canal, it gets coated with microbes from the mom. These microbes may kind of seed the baby with just the right mix. Combined with bacteria in breast milk and other microbes we encounter early on, they seem to slowly take shape in our first few years of life. The overall mix of our microbes becomes very personal, sort of like a fingerprint or maybe a blood type. But our microbes tend to resemble those of our parents and siblings and may stay with us for much of our lives. They may also be doing all sorts of things, such as educating our immune cells, like this one, teaching them the difference between things they should fight off, bad bugs that might make us sick, and things that aren't a threat, like our good microbes. When we're adults, microbes become our first line of defense, fighting off germs that try to invade our bodies, protecting their turf while protecting our health. Scientists have discovered they can even spew out their own antibiotics. The types of microbes in your body vary depending on exactly where they live, like different ecosystems in nature. There are wet places like our mouths, noses, and armpits, oily places like our scalps and backs, and dry places like our forearms. Different species of microbes have adapted to each of these habitats. The biggest, most important microbial habitat seems to be in the gut. It's the most complex, the most diverse, and everything microbes are doing everywhere else in our bodies, fighting off infections, revving up and dampening down our immune systems, signaling cells, that's all happening in the gut in spades. They even seem to help regulate our metabolisms, how much energy we burn, and how much fat we store. So if it's not functioning properly for some reason, because of what we eat, antibiotics we take, that may actually lead to all kinds of diseases. Diseases like colon cancer, colitis, maybe even diabetes and obesity. Some scientists think one reason a lot of diseases are increasing is because we've lost key gut microbes. Our microbiomes look far less diverse compared to those of people in less developed countries and earlier generations. And remember how we get our microbiomes in the first place? From our mothers when we're born and from breast milk? Well, some scientists think that too many babies aren't getting that because of all the C-sections and not enough breastfeeding, plus all the antibiotics kids get these days and our obsession with cleanliness. All this may help explain why problems like asthma and allergies have been soaring. Maybe because our microbiomes never taught our immune systems how to work the right way. Maybe swallowing good microbes, probiotics, could prevent and treat some diseases. So could taking prebiotics, essentially food that good microbes love. We end our story with a reminder. This research is really new. We still have a lot to learn about what many of our microbes are really doing. But scientists say that it's getting clearer and clearer that the tiny organisms all over our bodies are essential to our health and happiness.
So you can see that it really is a new area of medicine, a new area of science. And so, excuse me, what we're going to be looking at here today, and we are going to get onto a human microbiome after we take a look at cheese, but what we are going to be looking at here today is going to be an example of this very new area of of science. So let's move on with our presentation. Let me share my screen again with you so that you can see this. Right. So there are two aspects that we're going to be looking at as part of understanding microbiomes. The first is experimental, so how do you generate these data? And the second is computational, how do you analyze the data that have been generated? And we are going to be doing a computational analysis today in the hands-on session of data that were collected experimentally by other investigators. In fact, they were collected by students at UCL. So one of the essential aspects of microbiome data analysis is that you're looking at the DNA sequence of the microbes and their genomes. And DNA sequencing is something that has gone through several technological changes over time since it was first invented, and we'll look at some of the steps in this process. But these changes have made it far less expensive to do DNA sequencing at genome levels, both for humans and for microbes, for anything, than it was previously. So this is a very accessible technology. And you can see from the graph that the change in slope of the decrease of the cost came particularly when a new technology called high throughput sequencing became available and came into widespread use. So that's what enters the market means. It's not just a research and development technique that a few small groups use. This is something that's used everywhere, and there are companies who will sequence your genome for you for not very much money. So there are two different ways in which microbiome sequencing can be done. And these are represented with the 4 jigsaw puzzles below. So one method is called shotgun sequencing, and the second is called marker sequencing, marker gene sequencing. And in shotgun sequencing, what you do is you sequence every bit of every microbial genome. So the 4 pictures represent 4 different genomes, and each is sequenced in its entirety to be able to be assembled to give you the full picture. In marker gene sequencing, it is much less labor intensive because you do not sequence the whole genome. You sequence one very small, specific, conserved part of it. And that is shown in this example by the blue circle in the lower right-hand corner of each of the 4 pictures, which represent 4 different genomes. And you then compare those to each other. So if you look at the picture content that is within each of the 4 circles, you will be able to see where it is different to the other circles in a particular circle and where it's similar. And this kind of information is used to determine relatedness between the different genomes that you're looking at the marker genes for. And this then can be used to generate the kind of tree of relatedness that we saw when we looked at the phyla. So to be able to do marker gene sequencing, you need to pick the marker gene very carefully. It needs to be something that is expressed everywhere. So in all of the different bacterial or fungal or archaeal species that you will be looking at. And the best candidates for these are things called housekeeping genes. So if you think about the DNA in your cells and what happens when you need to make new cells, when a cell grows and divides, there are some typically conserved processes. You replicate your DNA to make a new copy for the daughter cells that will be produced after cell division. You transcribe bits of your DNA into RNA, and some of those bits of RNA will be translated into proteins. So replication and transcription and translation are key biological processes in every living cell that has a genome that divides. So there are going to be elements in there that are everywhere that need to be the same with not many changes. But you also need a marker gene to have some regions that are different to each other so that you can tell when something is different from something else by looking in that region. And you need to have regions that are the same so that you can actually take that region out and look at it and see what's in the variable region. So I'll explain that in this example. When we translate messenger RNAs into protein. This is done by translational machinery called a ribosome. And a ribosome has multiple subunits, and we are looking at the data for the large subunit and the small subunit. So these two subunits go together to make a complete ribosome. And ribosomes have protein and RNA in them. And this table shows you, and the figure underneath it shows you what the RNAs are that are associated with each of the subunits. And you can see that prokaryotes, like the bacteria we're interested in the microbiomes of, have a small subunit RNA called 16S, but eukaryotes have 18S. So if we use 16S as a marker gene, we will be able to understand what is happening specifically with the prokaryotes. And we won't have the noise of the eukaryotic information coming in. So 16S ribosomal RNAs tend to be used as marker genes when we do microbiome analyses. So RNAs are single-stranded molecules, but like the DNA double helix, which can go together, the two strands, to form the double helix through hydrogen bonding, single-stranded RNAs can have local regions of hydrogen bonding, and this is what we're looking at here. This is the 16S ribosomal RNA, and this is all the local hydrogen bonding that takes place to make these stem loop structures. And it's been divided and colored into different regions. So we have 6 different regions that are identified here, and some of them are conserved and some of them are variable. And you can see regions V1, V2, V3, all the way up to V6 indicated near each of the colors. So if you take the E. coli genome and the E. coli sequence as a reference, and on the x-axis, we see the position, the nucleotide position of the E. coli DNA, starting at 50 and going up to 1,350, laid out in a linear fashion. And that linear fashion of the number of the nucleotide sequences maps onto the linear fashion of the structure of the RNA here. And I'm going to see if I can grab my laser pointer here. So what we're looking at in this region, you can see it starts at 400, comes around to 420, 30, 40, 50, 460, 470. We're in V3 region. And as we come into this yellow, we're heading into V4 region. And this area is of particular interest to us. It's 515 to about 536. And there's another region here on the other side of V4. So V4 is all here in yellow. This other region is particularly interesting to us. And these two blue regions correspond to these two blue arrows. And the reason why they have been selected is because they're very highly conserved. So if you look at a whole range of different bacterial genomes and compare them to this E. coli as a reference, the larger the H prime number, the more variability there is at a particular position. So these large peaks are areas of great variability. And you can see that this is hypervariable or variable region 4. And the regions with the lowest H prime numbers are the regions with the highest amount of conservation. And so if you choose these regions, they are conserved best amongst all species, and so can be used to find all species. And you can then look at the DNA sequence of the variable region in between to see what different species you have. And this is the basis of the marker gene sequencing. And for this isolation, why not between V6 and V7? It just happens that you get a greater length when you, let's hop back and look. You get a much better length when you're looking at this V4. It gives you enough region to identify the differences and understand more about the different species than something that's a little bit shorter. There are some people who do 2 and 3 together. There are a number of different combinations that you can do for this, but we picked V4 simply because there's a Earth microbiome project that looks at all the different microbiomes and all the different ecological systems and collects these data as crowdsourcing. And there's a human microbiome project as well that's based on the Earth Microbiome Project. And both those large consortia projects we're using V4. So we can compare our data to a massive data set that other people have, and we can take information from other people's sequences and study it for ourselves if we pick something that's more like what other people do. So that's why we picked V4. So the reason, the way in which you amplify these things, I'm not sure if you're familiar with PCR or polymerase chain reaction. If you're fond of watching detective stories, murder mysteries, forensic movies, or anything of that kind, this is the technique that can find what specific DNA there is from a very small amount of starting material. And it does it through a process that has 3 steps. The first step separates the strands, the double-stranded DNA, with heat. The second step allows very specific primers, so like these guys, the 515F and the 806R, very specific primers to be annealed. And the third step does the amplification. So we separate, we anneal, we amplify, and you get doubling each time. So with each cycle of PCR you do, you get 2 more, times 2 the number of copies. So if you do 35 cycles, you have 2 to the power of 35 molecules resulting from the end of that. And this is something that won a Nobel Prize. So here's our primer sequences, the 515F and the 806R. These are the things that flank V4, and these are the things that we've used to pull out the different sequences you'll be analyzing. And you'll be familiar with DNA having 4 bases, GATC, in it, and you'll notice some unusual ones here. And this is where it could be a T or a C. A Y has been put in its place, and when we look up Y, we see that it's C or T. So these are synthetic pieces of DNA. They're made chemically, and they're made on a machine. So the first base will be put in a G, then a T, then a G. And then at this point, machine will put in either a T or a C. So you now have 2 different populations of molecules. And then it carries on, and then it comes to M, which is A or C. So you had 2 different molecules starting up here, and then a further 2 possibilities there. So you have 4 different oligonucleotides in this mix, all in equimolar proportions. And so this is what you are using as your forward primer for PCR. So remember, we have a primer here. This is your forward primer. So this is the 515F, and then the reverse primers on the back here. And this has an even greater level of, it's called degeneracy because it's not unique. There are multiple possibilities. So there's 4 positions here for N, 3 for V, so that's 12, and 2 for W. So there are 24 different possibilities here. And this is a way in which people can make a region that is highly conserved, but not perfectly conserved, have the best match possible for all of the molecules in the sample you're looking at. And this is just to show you how complicated these primers are. This is the sequence of the forward primer we're looking at. This is the bit, remember it had the Y and the M. All of the rest of this has to do with the sequencing. So the primer is multifunctional, and part of it relates to the experiment and what we want to amplify and look at. And the other part of it relates to the mechanics of the type of sequencing that we'll be doing. So just quickly, this data that you are going to be analyzing were collected on a MiSeq next generation DNA sequencer. This is a machine that's made by Illumina. Illumina have most of the market share in terms of next generation sequencing device. And this is a swish photograph from their website. And it doesn't really show you scale, but just for reference, this machine is somewhere between the size of your laptop and a small microwave. So it's very compact, and it can generate millions of sequences. So in the early days, I just want to give you a flavor for how technology changes things, because technology will change in the computing. We're working with Mare Nostrum today, which is a petascale machine just at the edge of exascale. We are working from moving towards exascale computing, which will enable us to do far more sophisticated things than we're able to do at present. So technological advances are important in being able to make significant scientific achievements. And I just want to take a look at sequencing with you. When people first did DNA sequencing, they used this version called chemical cleavage. The purines are A's and the G's, and one reaction cut purines. Another reaction was able to cut specifically G's. A third reaction could cut pyrimidines. Remember, we saw those as Y in the degenerate primer sequences, and a fourth reaction can cut C's. So if you look at this, these are all radioactively labeled with 32 phosphorus, not exactly the healthiest technique on the planet, and something that needs a great deal of care to make sure that you don't contaminate the area in which you are or yourself. If we look here, we can see that at the first position, there is a band or a signal in the pyrimidines and one in the C specifically. So we know that that's a C. And here there's a signal in the pyrimidines, but nothing in the C. So we know that's a T. And you can go through and read the sequence by the shortest molecules at the bottom towards the longest molecules at the top. Not only did this use radioactivity, some of the compounds used for cleavage were things like hydrazine, which is actually rocket fuel. So not only was it radioactive, it was explosive, and it was low throughput, and it was time to move on. So this is another Nobel Prize winning technology, Sanger sequencing, and it had a radioactive version and then was converted with the use of fluorescent molecules so that things could, instead of needing 4 separate reactions, could all be done in the one reaction and run out together. This kind of fluorescent detection uses the same technology that you have in your supermarket scanners. When somebody passes a barcode or a QR code across a scanner, it's picked up by this kind of thing. And this is an RNA base. It has a hydroxyl group at the 2 positions here. This is a standard kind of base that you find in DNA. It's called deoxy, and that's because it has no oxy, no hydroxyl there. So it's deoxy at this position. And when DNA is synthesized, this hydroxyl will attach to this phosphate on the incoming base. And this is how you make the chain that covalently links DNA together. Sanger sequencing is based on dideoxynucleotides. So it doesn't have a hydroxyl at this position, which you would expect because it's DNA, but it doesn't have it here either, which means that when this nucleotide is added to the chain, nothing can be added afterwards. It's chain termination. It's the last thing that goes in there. And if this last dideoxynucleotide was an A, then you see a green signal. If it was a T, you see a red signal, and you can see how the Sanger sequencing would work and be a significant improvement in throughput. So you can get more sequences in less time. And this is just a further description of it. You have this slide, you have these presentations in your information that you've copied across from Cheese Template. So I just wanted to give you an opportunity to look at this at your leisure, because this shows exactly how it's done and describes the detail. Next generation sequencing, this is the Illumina sequencing. There are a couple of different versions of it. We're going to look at this optical sequencing because the sequences that you've got to analyze have come from an Illumina MiSeq sequencer and its optical sequencing. So unlike the previous sequencing, which relied on terminating a chain, this is actually sequencing by synthesis. And it's all done on a flow cell that has millions of clusters, each of which are being sequenced. They're sequenced in clusters so that you have enough information to be able to detect a signal. And what happens is you have the template strand of DNA that you want to sequence, and you have the enzymes, DNA polymerases, and all of the nucleotides that are available to read this template strand and cause the synthesis of the new strand. And so if there's an A here, the DNA polymerase will put a T, an A gets a T, a C gets a G, and it goes all the way through like that. When each base is added one by one, there's a sort of a terminator or a blocker that's put on it, and then an optical snapshot is captured, a photo is captured of it, and then the next base is added, blocked, and detected. Next base added, blocked, and detected. And so if you follow this dot, you can see that it goes from green to blue to red to green to yellow, and you can see from the colors what that represents in sequencing. So this is how the sequencing data that you're looking at were generated. In terms of microbiomes, there are a number of different things that we can analyze, and Art and I will be going through those with you today. The taxonomy, remember that structure that we looked at from biota all the way down to species. The taxonomy is something that is very interesting. Here is an example. We have different samples along the bottom, and we have the relative frequency of the different phyla within them and an indication of what each is. And you can see that there can be great variation. Let's look at this sort of light purple one here. It's very significant in this sample and very, very small in this one. And it moves around a bit. And the balance between, these are Firmicutes bacteria in green, the balance between the different compositions, the different taxa, the different phyla that are in a sample is as important as the number of molecules in there. So diversity and richness are things we'll look at. And this taxonomy tells you what the relative frequency is. This is a way of looking at the tree phylogeny, making these trees of relatedness. So things like we saw on the other slide that are more closely related to each other will be branches on the tree and further away from things that are very different. We can also look at the diversity of the microbiome. So we have two measures of this, and we will be looking at how to analyze each of these. The alpha diversity is the richness within a single sample. So how many species are in there? What's the richness there? And the beta diversity is the similarity between samples. When it says distance here, it's the distance of how close is it to another sample on the tree. And what we're going to be using for this is a program called QIIME2. It's quantitative insights into microbial overview. And it takes these raw sequences that we're going to provide you with and puts it through a stepwise series of processes. It takes them apart and assembles them so that all the sequences from one sample are put together and able to be analyzed, joins up its pairs and deconvolutes that. And then it goes through and as part of its analysis generates the outcomes, the taxonomy, the phylogeny, and the diversity that we're interested in. And it gives a capacity to represent these and visualize the data. So do we have any questions before we move on to what we do next? I wanted you to understand the biological importance of the computational analysis that you're going to be doing today. So if anybody has questions, I'm perfectly happy to take them in the chat, or you can ask them as we go along, and we'll be keeping an eye on the chat and try and pick them up there. Are we all good? Shall we carry on? I'm going to assume silence means yes. So this is the end of this introduction, and we're going to head on and do the hands-on workshop now. So what I'm going to do is stop sharing my screen, and I'm going to hand over Art. Shall we kick off?
Hi, hello. Yes, let's go for it.
And can I just ask people quickly, could you put your hand up, please? Could you raise your hand if you have been unable, you could not copy the files across? Can we just have an indication? I'd like to know and be able to help the people who may be struggling with that. Is everybody good? I'm not seeing any hands, so I think we should be okay to go, Art. I think everybody has it.
Okay, awesome. Hello, everyone. So yeah, now we're going to do the hands-on walkthrough through QIIME2. And essentially what we're doing is we're analyzing a data set of which basically focuses on the microbial diversity of a few cheese samples. So the question is there a significant difference between the microbiome of pasteurized or non-pasteurized cheese, so treated or non-treated cheeses. So yeah, to start this, we're essentially going to have to activate the QIIME2 environment in our terminal first. So the first thing that we should do is type in these commands. So module load anaconda dash 2019.07, and then source activate QIIME2 dash 2020.11. Right, so if this is successful, you should see that QIIME2 pops up in the corner here, which basically indicates that we're ready to start using QIIME2 within this shell. So the first thing that we want to do here is just cd to our cheese directory that we copied over. And then in the cheese directory, you should be able to see each of these files. So we've got 1 cheesefilter.sh, 2 cheeseclassifier.sh, 3 cheeserareFraction.sh, and 4 cheesediversity.sh. So essentially what these are is they're pre-made scripts which I've created to essentially take you through this analysis pipeline, which is a typical kind of pipeline that we would use when conducting microbiome analysis in QIIME2. So if we take a look at some of these other files, these ones here, the alpha, beta matrix, dblur, these are the outputs of the analysis that we're going to do. So you guys don't have them in your directory just yet. But the files that you do have are, for example, we've got PE reads right here. So PE reads contains all of the sequence information that we're going to analyze. So PE stands for paired end, which basically means that, so as we discussed, we're taking samples using 2 primers. So the 515F primer basically is the forward read of this V4 hypervariable region, and the 806R primer is basically corresponds to the reverse read of the hypervariable region. So each sequence contains 2 reads that we're going to have to pair during the analysis. So yeah, so these are PE reads that we're going to analyze, and then we've also got these scripts. So if we take a look at the first script, we'll just type in vim cheesefilter.sh. Okay, so this is the first step within the analysis pipeline. So let's take a look at this script from the start. So this initial line essentially just tells the scheduler what sort of language we're going to be using within this script. So this tells it that it's a bash shell. And then here we have a bunch of sbatch commands, which essentially communicate to the scheduler the sort of resources that we're going to have to be allocated when we run this job. So here we're specifying the job name. Here we're specifying that we're going to need a maximum of 2 hours for the analysis to complete. This line here, the reservation, essentially we've been allocated a reservation for this session, so it's telling the system that it should prioritize our script for running the analysis. And then here we've got a quality of service training, which is another basically just telling it what type of service we need. And then N48 basically says to the service that we want to run on 48 cores on the system. Okay, so let's just jump straight in. So here, this is the first step. So in the first step, what we're doing is we're importing the data. So this is the sequence data that I discussed earlier that we're taking from the PE reads directory. So we've got an input path here, which is manifest.tsv, and this is a file which essentially just points the script to that PE reads directory to source those sequences. This input format basically just tells the program what kind of format our sequences are. So this is paired end reads FASTQ manifest Fred 33v2, which essentially means that they are FASTQ files. And FASTQ files, what they are is they've got a sequence, and then each base within the sequence is assigned a quality score. So during your sequencing through an algorithmic procedure, the sequencer will essentially assign to each base the probability that that base is actually correct and not incorrectly called. So for example, if we have a sequence ACTG, it could say that the A has a probability of 100% of being correct. So we wouldn't worry about that, and we would keep it within our sequence. Okay, so once we've imported the data, we basically output it to this file called dmux. So essentially, this creates a file called dmux.qzv. And in this next step, dmux.qza. And in this next step, we convert dmux.qza to dmux.qzv. So what a QZV file is it's a visualization file that essentially allows us to visualize information about the respective QZA file. So if we take a look at the dmux.qzv file for this analysis, when we visualize the dmux.qzv file, we see this. So if we scroll down, we see that we have each of the samples. So these are the sample IDs. So we see 515 RCBC282 has a sequence count of 1,366,712, right? Whereas we see that the sample 515 RCBC281 has a sequence count of only 2,873. So that's pretty interesting. This basically shows us how many sequences each sample has upon importing the data. So what we're going to do in the next steps, because this is raw data, is we're essentially going to process the data so that it's ready for analysis. So the first step of this is that we need to join the pairs. So as I said, we have a forward read and we have a reverse read for each sequence. So those forward and reverse reads need to be paired to one another. So essentially here, we're taking this dmux.qza file, which are our raw sequences, and we are converting them to a set of joined sequences. So in the next step, we again convert the joined sequences to a visualization of the joined sequences, which gives us this file right here. So now we see that the sample after joining the sequences with the most sequences is 515 RCBC288. And we see that the number of sequences for this has gone down to 801,387. And then for the one with the least sequences, we have 1,758 sequences. So that corresponds to 515 RCBC281, right? So now we've joined the sequences. So for each sequence just has one corresponding sequence. Okay, so the next step is to filter these sequences by the quality score. So as I mentioned, these sequences initially were in FASTQ format, which means that every base has associated to it a quality score, which corresponds to the probability that that base has been incorrectly called. So what this does is it basically takes a quality score threshold and it discards any sequences that contain a certain number of bases below a certain quality threshold. So essentially, this is the first stage at which we're actually starting to eliminate sequences based on the quality of the sequencing information, because we don't want any bad quality sequences in there, as they'll just introduce noise and noise into our data set, and also probably give us some incorrect information in our final analyses. So yeah, so again, we just convert this file that we create, which is the filtered.qza file, so filtered sequences.qza, to a QZV file. So when we visualize that QZV file, we see again that if we compare the numbers, we see that for this sample, for example, the number hasn't gone down at all, which is interesting. It means that all of the sequences, oh, it has gone down a bit actually. Yeah. So this sample has eliminated a few sequences due to the quality score of those sequences. And then, so yeah, we can see that the numbers are going down due to elimination based on quality score. Okay, so the next step in this script is the deblur step. So essentially the deblur algorithm is the final quality control step that we take within the analysis for our sequences. And what it does is it trims all the sequences to a standardized length of 253, which is roughly the length of the region that we're interested in. And it essentially classifies them based on which sequences are true biological variants. So these are called amplicon sequence variants. So if it deems that two sequences are different, but aren't sufficiently similar to belong to two different species, then it will keep one of those sequences, as those two sequences aren't true biological variants of one another. So essentially, this is reducing the noise in our data set and will increase the fidelity of our analysis. So at this step, we just visualize the statistics of the deblur, and it gives us the number of sequences that we're left with to conduct the final analysis on. So if we look at the deblur output, we see this table right here. So essentially, in this table, we see that we are ordering based on this reads hit reference column. So reads hit reference basically tells us how many sequences do we have remaining after this filtering step. So we see that the sample 515 RCBC 282 is the sample with the greatest number of sequences after this filtering step. So this sample has 394,214 sequences remaining after filtering with deblur, whereas 515 RCBC 281 has only 63 sequences remaining after this filtering step. So we see that there's a dramatic difference between the sample with the least sequences and the sample with the most sequences. All right, so basically this series of steps that we've done here, we've gone through from the raw sequences to a set of sequences that are standardized and correspond to true biological variants, and basically a set of sequences which have also been filtered for quality at this step. So this is generally the first stage of a microbiome analysis. And yeah, so I think it would be good if we ran the script. So to run the script, you should type in sbatch one_cheesefilter.sh.
So if people could stick their hand up, raise your hand if you're having difficulty running your script. What we'd like to do with you is to have everybody run each script at the same time, and we will Art will describe as he has what's in the first script and the visualizations, and then we'll run it. And while it's running, we'll look at the second. And when we have the output from the first, we'll move to the second, and we'll step through like that so that everybody can go through and obtain the sequence of things. Okay, so we have one. Can you put in the chat, please, what your difficulty is, and we'll try and sort you out.
Hello.
Hi.
Hello. Just one question, if it's okay, general question.
Of course, yes.
I was wondering, like, for example, today, I mean, afternoon, or maybe tomorrow topic, as I understood, every day is a different topic, like tomorrow is another topic, and yeah, maybe Friday is another topic. These what we will do will be a bit different from each other or something.
Well, what I was describing in the virtual human film, the vision that underlines what Comp Biomed is doing, is looking at the different aspects relevant to humans and their health and well-being, and it happens to be at different scales. What we're looking at today is the molecular scale. I understand you will be doing blood flow tomorrow and things after that, and you have a program that's quite nicely laid out that describes each of those things. What we're working on now specifically is the molecular level, and so we are best able to answer questions about the molecular level. But if you have questions about the program, you should message the organizers about that, and they'll be able to help you, because I don't know the rest of the program as well as I know this bit.
I see. Thank you.
So have people been able to launch their first script? I have one person who's having a bit of an issue, and we're sorting that out. But is everybody else okay, or are you just quiet? Because we have four.
Can I have one question?
Yes, of course.
I'm wondering, why do we need to activate GIME to 2011 in an active login node bash shell?
So Chime 2 is the program that we're using, and it's a Python package, and it runs in a Conda environment. And so we need to, well, with anything, you need to load the modules, and we need to activate this as a source.
Yeah, sure, but we are running it using the Slurm queuing system, not the active, our active shell. So why do we need to do this here?
I don't know specifically. I know it doesn't work if we don't.
Okay, thank you.
I mean, that's a very pragmatic answer. I don't know the details. We can ask support about this specifically. I know that we were having some problems activating it off Git Bash. We could
that's
only.
That's a good point, though. I thought the same myself, to be honest. But I did run a few scripts without doing it and tried it, but they just wouldn't run. So yeah, we just need to do it. I guess we don't
Yeah,
know exactly.
I'm afraid that's not deeply satisfying
Yeah.
as an answer for a mechanistic explanation, but it's a pragmatic one. And we'll try and find out the answer for the mechanistic one in the interim.
Okay, thank you.
You're welcome. Anybody from support online got an answer for this? Do chip in, please. We could use some help.
Hello.
Hello.
Hello. Yes. So I have a problem because I'm seeing the same thing what you are right now on the screen, but I am unable to, what I can say, execute the actions what we took last means before you.
Okay.
I don't know why.
Are you on a Windows machine?
I'm on
No,
Linux
I'm
right
on
now.
Mac. I'm on Mac right now.
Oh, sorry.
You're on Mac.
Which
So
part
here
exactly are you having difficulty with?
So right now I am seeing the same screen, means what you are seeing right now on the screen.
Yep.
I am at the same stage. But when I put the commands which you give us, I am not getting exactly what you did.
So which command exactly are you referring to? The sbatch command?
The thing is that when you type that anaconda kind of thing, then
Yeah.
there is something. Yes. So yeah, I can type for everyone this one. I will show you.
Okay.
So I just shared with you. You can see.
Hello. There was a problem with the sourcing the KIM environment that was related with LangC and LCO. So if you just do the export,
it's very difficult to hear you. You're breaking up.
Comments in the chat.
We're aware of a problem trying to source activate on a Windows machine from Git Bash. We're not aware of problems source activating from a Mac terminal. We're not aware of problems source activating from a Linux box. I don't believe there are problems source activating from a Windows machine running MOBA Xterm. Does that help answer your question? I'm sorry, I could not hear you very well. So if people have been able to submit this job using sbatch, if you type SQ, you should be able to see the progress of your run. The sbatch command, hang tight. It is sbatch. Oops. What is it?
It would be
sbatch 1_.
Yeah.
Let me grab it. Oops. So is everybody good? Have they managed to submit the first script?
Yes, I think now it's fine.
Okay. And when you submit that first script, you should be able to see the first folder pop up, which is joined, I believe, right?
Yeah, so the first file which is created is demux.qza. And then the first folder that's created is joined, yes.
I don't know if we can see other people on the queue. I don't know if we can see all the NCTs.
Yeah, I don't think we can.
Okay, so we're not able to see how you are doing because we don't see the whole queue. Ah, there's a message here about I am not getting these. You are not getting these files and folders? What do you get when you type SQ?
Hello.
Hello?
Yeah, so like when I opened, so I got an output text file. I mean, yeah, and when I opened that, there is a runtime error in it.
There's an error?
Yeah, runtime error.
Runtime error.
So you submitted sbatch 1_cheese_filter.sh.
Yes, yes.
And when you checked in the queue, had your job completed?
There was nothing in the queue, so I'm assuming it was completed. Like first something popped up and then it was empty.
I would resubmit it to the, how long does this one take to run? I can't remember. We ran them on the system yesterday and all these scripts worked on this system, but I can't.
This one takes about 40 minutes.
Right. So Amog, there's no way that this will have completed in that time. Could you resubmit it, please?
Yeah, sure.
And then follow its progress in the queue, because it should not complete that quickly. But you can also look to see, I don't know that we have the Slurm output file, which will contain errors as well. So if you cat the file, you should be able to see what it says. But my guess is it just hasn't completed yet.
Can I just post the error in the chat box?
Okay.
Yeah, sure.
Oh, yikes. Somebody saw something like this yesterday, didn't they? Wasn't there something about an ASCII 2 issue? I just remember seeing it.
Oh, yes, yes. This was the same issue that I had, Andrea.
Yes, okay. How do we get around this? Does everybody need to, does this need to be in the script? No. Let me just grab this.
I think Pierre might have a solution.
Okay.
He typed it in the chat.
Yeah, I've seen that. Is that in setting the environment? No, it goes into the script, doesn't it?
There are two options. One is that you have like a fresh environment, and then you do this like export this local stuff. Or the other possibility is that we add those things on the top of the sbatch shell scripts, just below the commands for sbatch.
What's the easiest?
I mean, the easiest probably is to just create another SSH session and start fresh.
Okay, because when we did this, we'd purged the environment. When I did it, I ran it with the Anaconda and BSC 1.0 were the only things that were in it. I'm not sure if, I mean, it runs okay like that. Should people just abort and come back in?
I believe so, but I haven't gotten errors because I started the sbatch commands from the wrong terminal with the error. So I
Oh, life's rich tapestry. Okay, why don't we start from scratch and people just log back on, and then we can do module purge. Okay, right, let's do this.
Okay, I'll go back and check.
Okay.
So the export commands, like, should we include it in the bash script below the sbatch commands?
I am not sure. I thought we were resetting the environment. Art, how did you sort this when you encountered it?
I encountered this on Windows, Charm GUI, and then I just switched my operating system, and it was fine. But I guess that's too drastic.
Should we put it into the script, into the batch script?
Yeah, we could do that.
Can you just open up the batch script and give people a walkthrough for that so that it's
yeah, sure. So, okay, so you just want to vim into the script. So vim one_cheese_filter.sh. And then if we just go to the top of the script, we want to insert these commands right here. So if you just click I, you will enter insert mode. And then we can type in the commands that have been given to us. So export lang equals c.utf-8, and then export lang, lc_4. So once we've got this, we press escape to leave insert mode. And then once we've pressed escape, we can type in :W, and this will save the changes that we've just made. And then to leave the script, we type in :Q!! And that completes that process. So that should fix the issue.
Right, so how is everybody doing? Have you managed to modify your batch script to include those commands, the export commands? And has everybody managed to submit their first script to the queue? So before we carry on, we really need to understand that everybody's got successfully to this first state. So I'm going to ask if people could raise their hands if their script is in the queue.
Excuse me, may I have a question?
Yes.
I think this user ST, I mean, I have PD as a status, I think.
Yeah.
I'm not sure what does it mean.
That means it's pending, so that's good. You're
Huh.
just
But I have
basically just
two.
been submitted.
But actually, I mean, I think I mistakenly I thought that it's like postponed or something. So I made another one. It has one priority and the other one is resources. How can I delete the other one?
So on the left-hand side, so
S cancel and the job number.
Yeah.
Okay. It's like a kill a command.
Yeah. You're just pulling it out of the queue.
Okay, great. Thank you very much.
Right, so I'm looking at hands in the queue. Who have we got? We've got a few people who have not got their script submitted to the queue. About half of you have not got your script submitted to the queue. So if you are having difficulty with editing your file or difficulty with submitting your file, could you please let us know in the chat or tell us verbally so that we can help you?
Basically, I have entered the script, but what I can say, I can't edit it or I can't go outside the script. I don't know why.
You have it in Vim, but you can't save it?
Yes, because I think it's stuck or something, because I can't get outside the script right now.
So if you click escape.
Yes, I tried escape and also what I can say.
Colon W. Art, could you maybe open it up?
Yeah. So if I can just type in, the commands into the chat and then.
Okay.
Yeah. So first you click escape, and then you can type in W and then Q like that.
Right. Okay. That's not come through. It might be a
Oh,
DM.
sorry. 1 second.
So does that help, Swapnil?
Now it is saying that /cheese filter sh return. So it's like this. It is giving me this message right now.
Could you type the message? Could you stick it in the chat so we can?
Yes,
I see. Okay, thank you. Well, that's the first script.
Yeah.
Yeah, so that's fine. So you need to quit now. You need to.
Now you just press :Q, so like this. And that should.
Now I think it's fine.
It worked?
Yeah.
Nice.
Thank you.
No problem.
And so what you want to type is the sbatch command now. Let me find it. All right, we still have some others who've not submitted. Is everybody okay, or do people need some help with this? So for this hands-on session, it's very important that we work out how to get everybody started for their first script, because the subsequent scripts are all dependent on the output from the first one. So we're really quite keen to make sure that we can get everybody able to edit and submit the first script. Okay.
Hello.
Hello.
Yeah. So Sorry for interruption, but I kind of joined a bit late, so I'm not sure whether I should just stand by, or maybe I need to run this anyways, and then I can try to send it later.
Well, have you found the information about copying everything across to your folder?
And I'm pretty I'm pretty sure I can do that.
Right, but if you've not found it, let me find it for you so that you can do that now.
Okay.
Somewhere. Hang on, it's way back in the thread. I guess when you join, you don't see what was in the thread prior to joining, is that right?
Yes, that's correct, ma'am.
Okay, gotcha. Well, people have had some complications in what they've stuck up here, but let me find what I was suggesting that people copy. Okay, here you go. Here is what you need to put to bring things from your home directory. So you just type cd return and make sure that you're in your home directory, and then just type that line that I've just put in, and that will copy everything across.
Understood. Wow, I'm having a hard time actually connecting. I'm doing the SSH, but I'm not connecting.
So you.
so yeah.
What
So
are
I'm
you?
doing SS. Yes, ma'am. Go ahead.
What are you connecting from? Are you on a Mac, a Linux, or Windows?
I am on a WSL2 on a Windows 10. So it's a Linux subsystem on Windows, but it should be fine. It worked yesterday. I was able to connect to.
So you type your account number, NCT thingy, what's it, at mn1.bsc.es, and you cannot get on like that?
Yeah, I did the SSH and NCT, and then my, the 01, and then my holder, which is 038 at mn1 at bsc.es. I mean, .bsc.es. Yep. Now I'm supposed just to put my NCT 01038, and I do that. I'm having a hard time though to connect.
Have you typed? Do you see the Mare Nostrum main screen? Have you typed your password in?
Yeah, yes, ma'am, I did. So I don't want to hold you. So I got to the command. I don't want to hold my colleagues. I don't want to be interruption.
No,
so yeah.
Don't worry, this is a 40-minute script runtime. We've got plenty of time to sort this out. So yeah, just relax, get on the system, have take a deep breath.
Okay, thank you.
You're welcome.
That makes me feel much better. Yeah, so I'm told the permission denied.
In general, that's in my experience when I've typed the password wrong.
Okay, I'll read that one.
I mean, I can touch type, but when I put passwords in, I tend to use 2 fingers, maybe 1.
Yeah.
Just to make sure I get it right.
Okay.
And stress won't help, so just relax.
Okay. I just woke up, like an hour ago. It's 4 a.m. here at US.
4 a.m. That's impressive. Which part of the US?
I'm in Pennsylvania.
In where? Sorry.
Pennsylvania.
Excellent. Right. Okay. So how are we doing here? So Swapnil, you've got 2 jobs that are running. So I'm not sure whether pending, there's a line in the script that indicates the reservation status that should be preferred. So it should not be pending. I don't know why this is. What do you mean priority at the bottom? You mean the priority of your run is at the bottom, or the priority is listed at the bottom?
So like when I type SQ, there is a priority in brackets at the left hand side.
Can you put in the chat what it says?
Yeah, sure.
Thank you.
Yeah, and
sorry,
Swapnil, if you have two scripts running, I think it would be best to cancel one of them. So
how
that you
can I
don't.
do that?
So you would type in S cancel and then the number of the job ID. So you would want to cancel the one that's pending.
176. So yeah, you want to do. Oops.
I should write yes, space, cancel, and the number.
Yeah.
Not space. So just like on my screen. So S cancel like that, and then the number.
Yes, cancel and the number. Continuous or with space?
Space between the command and the job number.
Yep.
Otherwise, it sees it as one command and it doesn't understand it because it's not the right syntax. Excellent. We have a laptop reboot. I'm not sure how much influence the environment has on some of these things. And I also don't know what impact being on the different terminals or systems coming at things from has. Seems to make a difference. Has anybody, okay, so I think we started submitting things maybe what, 15 minutes ago, Art?
Yeah, roughly.
Okay, so it will run for some time yet before it completes.
Yep.
And I think I would like to see if the people who have not yet submitted, start time estimate is 13:15. What's that? That's 12:15 here. Really?
You know.
Yep.
If these take a long time to upload, to submit, I think we could just go through each of the scripts and the outputs and the explanations.
I think so. Do we need to put the export command into each of the subsequent ones?
I'm not sure actually, because it would have, I feel like it would have configured the environment in that way. So like you don't have to do, for example, source activate chime2 more than once, you know?
Yeah, that's true.
But I'm not sure.
Sorry, somebody's trying to say something? No? Okay. We can't hear you very well.
I have a comment about the environments within a bash script. If you export it in the script, they're going to be active only
for the script.
Okay.
Yes.
Okay, so we'll put it into each script. So Art, maybe what we can do now is step through each of the subsequent ones, and we can edit them at that time to put that. Oops, that's the wrong one. I'll find it again. To put that information about the export path into each of them. And what we can do is ask everybody to open their scripts and look at them and make the edit at the same time when we go through them, so that will allow you to have each of your scripts edited in real time when we're doing this, and then they can be submitted sequentially. Sound okay for people?
Okay, should we
Yep.
start running through this then? Cool.
Yep, let's go. Vim.
Okay. So let's just edit the script first. So we're looking at the second script right now. Export lang equals c.utf 8.
So I hope that everybody's making these edits on their copies in their local files from what you've copied across from the projects, the GPFS projects folder. I hope you're editing these now in your own local directory, your own home directory.
Okay, so
I have one question. Question. How can I edit the script? Because when I type the enter button, it is not going down as I can see on your screen.
Okay, you have to press I first. So if you just press the letter I, it will enter insert mode. And then you'll be able to edit it.
So when people did the introduction yesterday with Okba and Carlos, did you cover editors? No? Okay. So we're using an editor. Yes, you did. Okay. Well, this is just the information. Yeah, VI or Vim. We are using Vim here. People can use whatever they like as long as it works for them.
So even now I'm not unable to insert anything. When I click I, it is inserting anywhere. So I don't know.
Art, could you exit out of that, save it, exit out of it, and then go back into it again so that we can just sort of do a walkthrough, please?
Yes,
okay?
Now I can.
Right. Okay. So Vim is the editor. You type the file name and then hit return, and you will see the screen that Art will see now. And at this point, you can look at it and scroll through it, but you can't edit it. You need to type I, the letter I, to be able to edit it. And when you do, you see in the lower left hand of the window, you see it says insert. And at that point, you can type into it.
Yes. I am able to click.
So Art has edited this script now to include the two export aspects that need to be put in there. And I think maybe best to write and then go through it and then quit.
Yep. So once you type in, once you make these changes, you would press escape, and then in the lower left-hand corner, you'll see that the insert mode has gone. So we're no longer in insert mode. And then to save the changes, we type in :W, which basically stands for write. So write changes. And it will give you this output. So it tells you that we've written the file. And then to quit, we do :Q. And that would bring us back.
Basically the colon is the instruction, the command instruction from within the editor. Is everybody good with this? Have we edited the second script?
So I have edited it, and now I should press what to execute it?
Nothing, because you need the first script to run first.
Okay, so now I just need to get out of this.
Yes. And Art will go back into it, and we've he's described what the first script did, which is basically get all the way through getting the noise out of the data set and getting the valid sequences ready to analyze. And we can now go through with you what happens in the second script.
Yep, okay. So the second script, the whole point of this script essentially is to take these filtered sequences, these denoised sequences that we've created using the first script, and classify them into taxonomic groups. So the output would be, for each sample, we'll have essentially the taxonomic diversity of the sample. So we'll see this when we visualize the output of this script later on. So the first step that we do here is we are essentially unpacking this file, gg_13_5 otus.tar.gz. And what this file is it basically contains, it's a reference database of 16S rRNA sequences, right? So it contains a reference database of sequences and their associated taxonomies. So for each sequence within the database, it has associated to it a taxonomy. So the taxonomy would tell you the taxonomic classification of that corresponds to that sequence. So essentially the species that corresponds to that specific sequence. So we're importing these reference reads. And the reason why we're importing the reads is because, so we have a bunch of genetic sequences and their associated taxonomies. And the reason why we're importing these is so that we can now train a classifier. So we're training a naive Bayes classifier, which is a type of machine learning algorithm, to essentially be able to classify the taxonomy of a sequence based on the content of that sequence. So yeah, so we're importing this data in this step with QIIME tools import, and then we're trimming each of the reference reads, and we're basically doing this process called in silico PCR by taking the forward primer and the reverse primer that we're using in our experiment to isolate from the reference database the V4 region of the 16S ribosomal RNA, right? Then once we've isolated that V4 region of the ribosomal RNA, we are feeding it into this naive Bayes classifier. So you see we have reference reads here and the reference taxonomy. So each read is associated with a taxonomy. So we're feeding this data into the naive Bayes classifier, and the output of this is a trained classifier that will basically be able to classify the sequences in our data into their respective taxonomies. So the next step, which says assigning taxonomy sklearn, is basically using this classifier that we just trained to do what I just described, which is classify our sequences into their respective taxonomic classifications. And the next step right here, creating taxa bar plots, basically it's creating the visualization of this classification. So yeah, so we can have a look at this visualization and actually see the taxonomic contents of each of our samples, which is pretty cool. So if we go to level 2, we'll be able to see the phyla that are present in each sample and their associated relative abundances. So for example, here we're sorting based on proteobacteria. So the samples here are are sorted according to the abundance of proteobacteria within those samples. So we see that sample 515 RCBC276 has the greatest abundance of proteobacteria with an abundance of 97.916% of proteobacteria in that sample. Whereas RCBC286 has a very low abundance of proteobacteria with only 0.459% in that sample. Now we can also sort based on the index and look at, for example, taxonomic level 6, which gives us the genus. So we see here we have kingdom, phylum, class, family, genus. And we can, for example, classify by index and see what genus is of highest abundance, for example, within this sample, which is 515 RCBC271. So the genus with the highest abundance in this sample is Lactobacillus at 32.419%. So yeah, that pretty much sums up the taxonomic classification that we can conduct using this taxonomic analysis that we can conduct using this script.
So if you remember back to the introduction that I provided, where I showed you the things that you can look at, the taxonomy, the phylogeny, and the diversity, this is this first step of the taxonomy, and it is in this second file that we will be generating those data.
Excuse me, ma'am.
Yep.
Yeah, I believe I'm in right now. I did run the command that you gave me, but unfortunately it doesn't go through.
Which command are you referring to? We've given several. Could you put it in the chat, please, so I know what you're talking about specifically?
Yes, ma'am. Yes.
Thank you.
The one that you, okay, I'm posted it in the chat.
Ah, that one.
Yeah, and I'm getting
and are you in your home directory? If you type cd and return.
Yes. Yes, this is where I am. I believe I am in my home directory.
I don't understand the difficulty because this is what others have typed to copy things over. There were some variations where people set themselves different folders to make it easier for themselves. Let me see if I can find one of those.
Okay.
Yes, the Green Genes database, Pierre, it's the reference data. It's the reference database that's used to train the classifier. And that is then applied to the data from the samples. You need some form of a training set for it. And you don't want to take and have your training set come from your samples. This is why you use a reference database, because if your training set comes from the samples, you reduce the number of samples that go into the test set, so you get less rich information back. So is everybody comfortable that we now have been working our way through the second script? And when the first script has been submitted, we will be in a position to submit the second script. It does look as though it's taking longer for these to run than we found when we ran them, and it may not be possible to finish in this session, to have the complete output. But what I would like to do is make sure that because this is being recorded, we go through each of these steps with you, and we go through each of these files, or scripts rather, to look at what is being done, and you will be able to review the recording and submit the next stage scripts when each run completes for you. So are we finished with our second script, Art?
Yes. So yeah, that concludes the last script. Well, actually, there's an additional step here that we didn't discuss, which is that the final step in the script essentially creates a phylogenetic tree for each of the sequences. So the data that's created by this step will be used subsequently in the diversity analyses, where we're interested in the distribution of phyla within each sample, or in other words, the alpha diversity of each sample and the base diversity between the samples.
So to get out of the script, we do the colon, the Q, and the exclamation mark, because we've been using Vim as an editor here, but you would exit saving your changes with whatever editor you chose to use, if it's VI or if it's Nano or what have you. We would take a look now, I think, at the third script and make the edits to that. So you should please make these edits along with us at the same time. That's the second one. Yeah, those ones. Gotcha.
All right. So again, we press I to insert. And then you want to type in these lines again. And then once you're done typing those lines, you press escape to exit insert mode. And then :W to save the changes.
And do we have visualizations from this that we would want to look at now, or should we look at everything at the end?
Yeah, we do. So, yep.
So you can see from each of these scripts that they are executing calculations that result in data output that can be visualized. And we're looking at the visualizations for these. And you can also see from each of them that they call files that are in the cheese template information that you copied across. So this one, for example, is calling metadata.tsv. So some of these scripts call similar things all the way through because they're associating the samples and the data associated with the samples with the output and the visualization of the output. You might find it helpful to open all 4 scripts simultaneously with the first one on the left and the fourth one on the right in sequential order, and look through them and understand the flow of what each of the things is calling and what each particular calculation is doing and how that adds up to the output we will come and look at the end. Has everybody been able to, if I could just ask people to put their hands down for a moment so that we can ask you a new question. I don't think we have a capacity to poll in here very easily, but I'd actually, let me have a hunt and see if we can poll. Can we poll? No, I don't think we can poll in Zoom, so that's less easy. Could I ask people to raise their hands if they have completed the edit of the third script? So you now have 3 scripts edited and 1 batch job submitted. Okay, excellent. So we have people who are following along well. And for those of you who are not at that stage or who have wished to simply observe rather than do this in real time as we do it, because you will do it later, you will have a good template through this recording for being able to see how each of these edits was made, see what each of the changes is and how each of the visualizations have been done. Should we move to the fourth script, Art?
Yeah, sure. Or I can explain this.
Oh yeah, of course.
What it's doing. Okay, so this step is an essential step that we conduct before basically conducting the final diversity analyses, which will give us an idea of the diversity, the microbial diversity of each of our samples. So what the purpose here is to find out how many sequences we want to sample from each of our samples within the analysis. So to get an idea of what I mean, let's just look at this table right here quickly. So within this table, this read-sit reference column is basically telling us how many sequences we have per sample, right? So this sample here, 515 RCBC282, has 394,214 sequences, whereas we also have a sample, RCBC281, which only has 63 sequences. Now, when we conduct the alpha and beta diversity analysis, we want to take a certain number of sequences from each sample to basically compute what the diversity of that sample is. But the catch is that we can only select one value for one sampling depth value for all of our samples. So, for example, you can imagine that if we chose a sampling depth of 63, right, then we would only sample 63 out of 394,214 in this sample, which is RCBC282. And what this means is that we would capture hardly any of the actual microbial diversity, which is represented by these 394,214 sequences. However, if we chose a sampling depth of 394,214, then the only sample which would be considered within our alpha and beta diversity analysis would be this one, 282, since all of the samples below that number get excluded within our analysis. So essentially, what we need to do is select a sampling depth that gives us a balance between basically understanding the microbial diversity of each sample and not neglecting too many samples. So generally, we would say that a sampling depth of 10,000 and above should be sufficient to gain a good idea of the microbial diversity of a certain sample. So in this case, we could, for example, say that we want to pick a sampling depth of 14,910 to retain all of these samples, but then discard these ones down here. And we could say that that would be the most appropriate choice for this data set, as it would give us a good balance between losing a minimal number of samples and gaining a good sampling depth to understand the microbial diversity of our data set. So basically what this step here does is it shows us how the estimate of microbial diversity changes as we increase the sequencing depth. So we see that if we have a sequencing or sampling depth of 2,500, then the diversity, this is phase phylogenetic diversity, which is a measure of alpha diversity, is much lower than if we select a sequencing depth of, for example, 15,000, right? So we see that the greater the sequencing depth that we have, the greater the diversity that is calculated for that sample. So yeah, so this visualization basically allows us to conclude that a sampling depth of roughly 14,000 would be good for this data set. So yeah, that's what the purpose of this script is. So moving on to the fourth script. So yeah, in the fourth script, we see here that we've selected this sampling depth based on the rarefaction step of 14,910, and that's basically the sampling depth that we're going to use to calculate our alpha and beta diversity metrics within our analysis. So before I go ahead with that, we should edit the script as we did before. So by clicking I and then typing these commands in so that it can run.
So hopefully everybody has managed to do the edits for this fourth script now. Could you please put in the chat if you have wanted to edit but not yet been able to edit this script so that we can help you out and carry on going through what the purpose of this script is and what outputs it generates? I assume if we don't hear from anybody either through audio or in the chat that we're all good to go with the edits of this script and then can carry on. Just edited once. Each of the four scripts needs to have the same export commands inserted into it. Swapnil?
Ma'am, I'm trying, but what is happening when I press for what I can say, the script file, it is showing something blank. So I don't know what happened. There is nothing there.
If the script file is showing something blank, I think you are not in the right path, because you can give a vim command and a file name. And if you are in a directory that has that file name, Vim will open it. If you are not in a directory that has a file of that name, Vim will create it and it will be blank.
Basically, I am in that particular file because I'm working on the first, what I can say, the first command. But for the next 2 and 3, I don't know why it is happening.
If you exit out of the editor without saving the changes and type pwd and have a look at the path you're in.
pwd, yes. So
you should be in your home directory, and if you type ls -l or -lt or -ltr or whatever
it is showing everything. It is showing everything like 1, 2, 3, 4 cheese, filter, classifier.
Yeah. Okay. Well, if you type so you have edited the first one and submitted it, right? So
Yes.
you are working now on the second one, is that correct?
Yes.
Okay, so if you type vim space 2 underscore and then that file name and open it up, it should not be empty.
And the file name will be the same, right?
The file name is the same as the name of the file as you see it in your directory, because it is what you copied over from the GPFS projects folder.
Yes, yes. So vim2 /g's filter sh.
Let me just have a look.
It's showing the same blank thing for me.
It should not be blank because that is a file that has it's 2 underscore cheese underscore classify.sh, yes?
Yes. I don't know why it is blank. Because I'm typing vim2 underscore cheese underscore filter dot sh.
There's a space between the vim command and the file name. Have you put the
Yes.
space? Okay.
Yeah, it's space.
If you forgot to edit the first and the second scripts, I think I would go back, edit them, and resubmit them. Not everybody seems to be having difficulty with the export. So it may be that you're on a system, Shabnam, that doesn't have a problem with export, in which case you'll be okay. But if you get the output from your first scripts and you have from your first script and you have an error in it, you will wish to edit the first and the second batch scripts and resubmit the first and the second. Could I just ask people if they could put their hands down for a moment, because I would like to ask a different question. And what I would like to ask you is, if you type SQ, has your job for the first script finished? And if your job has finished, if you could put your hand back up, please, that would be very helpful. Has anybody's job finished, the first batch job? So does that mean the hands that are up now, that your first job script is finished? Can somebody just sort of verbally tell me yes, so I feel a little more comfortable about this? Yes, excellent. Okay, so we can submit the second ones now. And that's what we should be doing here. And so what you want to do is type what I'm putting in the window here. Oh, you finished the second as well. Have other people finished the second? Are we on to the... Are you talking about editing or running, Shabnam? Running, excellent. Have other people submitted and completed the second script? So if you have completed your first script and are ready to submit the second one, let me just figure out where I am in this. You want to be typing the following command. So this is to submit the second job script. If your second job script has already been submitted and completed, then you would do the same thing to submit the third one. Has anybody had their third script complete? We have some who've had the second ones complete. Okay, so it's in priority. So the second one's running through. Has anybody, we have one person who has a second script finished. Has anybody else got a second script finished? No? Okay. Thank you for indicating that. Third one is running, second one's taking a long time to start. That could be the case. I'm not sure how the resources are being allocated on the machines, the machine at the moment, but I do know that these jobs are running through with priority. So once it starts, it should be okay. Art, how long did it take your second script to run when you ran?
About half an hour, no, like 25 minutes.
Okay, and the third and the fourth, I'm just going to make a note of this. So the first one was about 40, the second one was about 25 to 30. What was the third?
The third was maybe like 2 minutes.
Oh, excellent. Things are looking up. Was the fourth even faster?
Yeah, the fourth was maybe 3-4 minutes.
Okay.
Yeah.
So it's really getting past the first and second ones, and then things will be
those are
easier.
definitely the longest.
So if we could just submit as scripts complete, submit the next batch script so that you can step through with these, and you will be able also to do the same visualizations that Art's been doing. And perhaps we could look at the fourth one now for people.
Cool. Okay, so in the fourth script, we're calculating the alpha and beta diversity of each of the samples. We're doing this by taking a sampling depth of 14,910, which means that from each sample, we are sampling 14,910 sequences and then basically calculating the diversity of these sequences. So this step, just this one command, essentially generates all of the different alpha and beta diversity metrics that you would be interested in and outputs them to this directory, which is alpha beta metrics. So once this step is done, it essentially has created loads of different QZA files, which correspond to each of the different alpha and beta diversity metrics. So for the next step, we literally just generate the visualizations that are associated to each of these QZA files or associated to each of the different alpha and beta diversity metrics. So the first visual that we generate is for Faith's phylogenetic diversity significance. So basically what this tests is how different is the phylogenetic diversity of the different samples, right? So, for example, the way that phylogenetic diversity is measured is it's measured as the total sum of the branch lengths within a phylogenetic tree. So if you have loads of species that are very distantly related, then the sum of the branch lengths, because they will be distant from one another on the phylogenetic tree, will be very large. Whereas if you have the same number of species that are closely related, the sum of the branch lengths will actually be less, even though you have the same number of species. So we're not only accounting for the number of different species, but we're also accounting in this metric for the phylogenetic distance of the species within that sample. So yeah, this visual will essentially show us how different is the phylogenetic diversity of one sample or one sample group compared to another. So if we take a look at this, we see here that we can actually choose what kind of way we want to classify these samples. So we can classify it based on the batch size, based on the sample site. So we see that this is subsurface or surface. So this column corresponds to all samples, so n equals 7. So 7 samples that were taken from the core of the cheese, and then 7 samples that were taken from the surface of the cheese. We can look at starter material treated. So this basically tells us how the phylogenetic diversity differs between samples that haven't been treated and samples that have been treated. Or we can classify based on sample softness, so the softness of the cheese. Okay, so the question really that we're asking within this analysis is, is the microbial composition between treated and non-treated cheeses different, right? So we would select this column right here, starter material treated. And if we scroll down, we see the p-value. So the p-value of this comparison is 0.018, which basically indicates that there is a significant difference between the phylogenetic diversity of samples that have been treated with the process of pasteurization and samples that haven't been treated with the process of pasteurization. So yeah, that's one example of sorry.
So what's about that outlier up there? So is there a way we can, if we wish to eliminate it from the analysis in the script? I mean,
that's a good question. So I'm quite sure that through some process you could eliminate it from the analysis, but I think this points out that there is an outlier, but still the statistical test tells you that the difference is still different.
As a general rule, when we look at experimental data and we see outliers, we try to understand where they come from, whether they come from something technical, whether they reflect a rare event that if we increased our sample size, we would see more of. So as a general rule, you tend not to eliminate data points because that's introducing a bias that reinforces a hypothesis that you may be working towards. So as Art says, even though we see in the data that return here, the presence of the outlier, it's accounted for in the statistics and the p-value and the q-value. So generally you leave these things alone, but you note them and try to understand where they come from. And you always ask yourself if having more samples, larger sample size, more sequences. So for example, when we were looking at the sequences and there were 801,000 or some such for one and something like 1,700 for the other, you would obviously take that second sample and sequence it many more times until it got up around the 800,000 mark. So you try to understand your data and you tend to add data rather than subtract, because adding data enhances richness, subtracting reinforces bias.
Understood. Thank you so much. Thank you.
You're welcome.
Okay, so yeah, so that's an example of an alpha diversity metric where we're comparing the diversity, the within sample diversity between sample types. Now, Shannon group significance, evenness group significance, these are just different metrics of alpha diversity that account for different features. For example, some of these won't account for phylogenetic distance, but will only account for the number of different species. Some of them will account for abundance, other ones won't. So depending on what you're interested in, you can take a look at different types of alpha diversity. Now, the next type of diversity that we're interested in is beta diversity, which is essentially how different other phylogenetic groups between different samples, right? So how different is the phylogenetic distribution in one sample compared to another? It's a direct comparison, on the contrary to alpha diversity, where we're looking at the difference within the sample itself. So if we look at weighted unifrac is a way of testing this between sample diversity. We conduct a pairwise Permanova test and compare the samples that have been treated with the samples that haven't been treated and get a p-value of 0.288. So what this tells us basically is that the phylogenetic distribution between samples that have been treated and haven't been treated isn't that different. So basically, whether you treat your samples or not, the phylogenetic distribution won't vary too much. So it's quite an interesting result, really, because we find that the alpha diversity, so the within sample diversity, is significantly different, whereas the actual phylogenetic distribution between the samples aren't that different, if that makes sense. Yeah, so that's the beta diversity. Another way that we can visualize the beta diversity is through a principal coordinate analysis plot, which basically reduces the dimensionality of the data so that the principal components of variation are represented by each of these three axes. So axis 1 is the principal component of the variation in the data set, axis 2 is the second component, and axis 3 is the third component. So in these sorts of maps, we essentially look for clustering. So if we see clustering based on type, then we could conclude that that they are sufficiently different. So basically, points in this space that are close to one another correspond to points that are close in terms of their phylogenetic diversity. So here we've classified based on starter material treated. And if we look along each of these axes, so axes 1 and axes 2, or axes 3 and axes 1, or axes 2 and axes 3, we can see that the points are quite dispersed and mix with one another. For example, here we can see that these blue points and these red points are in quite a similar position in space, and these blue points are quite distant from these blue points over here. And this basically matches quite well with the with this analysis here, which showed that they're not the samples that are treated and the samples that aren't significantly different from one another based on their phylogenetic composition. So yeah, that's an example of beta diversity. And the weighted unifrac metric is, again, just one example of a type of beta diversity metric. So the weighted unifrac metric takes into account the difference in the phylogenetic composition, but also the difference in the relative abundance of each phyla. Whereas the unweighted unifrac metric doesn't weight the analysis for abundance. So this just straight up compares how different the phylogenetic constitution of each of the data sets is without taking into account the relative abundance of the phyla. Bray, Curtis, and Jakarta are just two other examples of this type of beta diversity metric. So yeah, that concludes the last part of this analysis, and this step is generally the last kind of step that you would do in a typical microome analysis experiment, since it allows us to answer the fundamental questions which we are asking.
So that's fantastic, Art. Thank you for that. I think what it might help to do is for me to share my screen and to show you a couple of pieces of information that I think that you will find helpful when you want to work through these things. I'm just trying to get them loaded up now, which is proving exciting, a bit more excitement than we needed. And let me just get the other one of these. So let's just look at these in, I'm trying to organize this so that it makes sense when you are reviewing it through a recording afterwards. So just really quickly here, what we have been looking at in this session today is an example of environmental samples that were...
Andrea, we are not seeing anything.
I know, I'm getting
Andrea,
there.
we are not seeing your screen.
No, I know.
Okay.
I'm just talking for a moment while I sort this out. So we've been looking at the cheese samples, and what I need to get the Zoom window off the thing here so that you can actually see what I'm looking at. Let me just try this again because I'm trying to share a screen. I've got two monitors and it's getting confused about which it wants to look at. There we go. Okay, so can you see this screen now? It has the two PDF files. You should be able to see these two PDF files. Is this visible to people?
Yes.
Okay, so this one on the left here is the description of the data set that we're looking at. And this was an experiment that was conducted to take a look at the properties of artisanal cheese and of regular supermarket cheese, and taking a look at pasteurized and raw milk samples. And so the barcodes, the sample barcodes, are now here associated with the samples from cheeses. And if you look at the metadata file, you will also see or be able to see how this information has been coded into the metadata file. So these are all the samples, these particular barcodes that came from the raw cheese. These are the samples that came from pasteurized milk. There were a number of different cheeses that were sampled, and they also, in this experiment, took a look at the region where things were sampled from. So some were sampled from the core of the cheese and some were sampled from the rind of the cheese. And then each of these things was amplified with a barcode. And you can see what the barcodes are in the barcodes.tsv file that you have in the information that you brought across. And the main question that was being asked was, is there a significant difference between cheeses that are made from pasteurized and non-pasteurized milk? Now, as Art was going through the walkthrough and explaining the different parameters and looking at the different visualizations, every now and then I popped something into the chat saying, this is one of the questions. So this is the kind of data analysis that you should be looking to be able to answer when you go through your analyses and you've completed all 4 of the scripts they've all run. And these are the 3 key questions that we thought would help you understand how to interrogate your data. And they look at different steps of it. So this first question asks you to look at the sequence data. What is present, you know, which samples have the most and which have the fewest sequences in the imported data. And then after subsequent steps, excuse me, including the deblur step, which just reduces down the noise at the end before you proceed to the analyses. The second is on taxonomy. So the first part of it asks you which is the sample that has the highest abundance of a particular phylum of bacteria, and asks you about the percentage. So that's using this bacterium with the greatest number and associating the sample with it. And this looks the other way around and looks at a particular sample and says, what is the most abundant thing within it? And then it asks basically for you to use the diversity metrics, which you have seen from Art's description of the 4th script, to answer the question of whether there's a significant difference, which includes the discussion about liars. If you wish to do that, what would you choose and how? So I'm going to stop sharing this for a moment, and I'm going to go back to my terminal and just show this to you in a moment. Let me just get back where I wish to be before I show you. Right, here we go. Let me find this screen and share this screen with you. Where's it gone? Here's it gone. Desktop 2. Can you see my terminal? Yes? Are you able to see this on this screen? You should be seeing.
Yes, yes.
Okay, cool. Let me make it a bit bigger so people can see it a little bit more easily. So this is the cheese template folder that you carried across, copied across. If we look at the PE reads folder, you can see each of these different FASTQ files that is present here. This is the primer number 515 reverse complement, and this is the sample number. So these are the samples that associate with the information. If we take a look at some of the other things in this file, we can look at the metadata. Whoops. And so you can see the barcodes for the metadata, you can see the specific barcodes they had, and you can see the variables for sample number and whether it was large or small batch, what surface. You need to map this with the information in the description to help you understand what you're looking at here. What else are we going to look at? The manifest. You should always take a look at the files that you have. Just cat them and take a look and see what they are. And this manifest is basically telling you how to put together the paired end reads. So for this sample 271, it's taking read 1 from 271 and putting it with read 2 from 271. So this is the paired end reads. What else have we got in here? Just so that you have a feeling for what you're looking at. We've looked at, well, the barcodes will just give you barcode sequence, but that's always, it's important to understand what your data looks like when you're starting an analysis, making sure everything's complete. So I can see all the barcodes that are in this experiment. I can see the sample ID, the specific sequence associated with them, and these are pieces of information that's used by QIIME to do what it needs to do. What else are we missing here? We are, what's in CheeseSH? It'll be a batch script. Okay, we're going to just avoid that for the second, because that's looking at script 4 and I'm not quite sure what that's doing there. And I want to point out for you this information folder. So in this information folder, you'll find the introduction PDF. So this is the presentation I gave you. You will find the description and the results worksheet that I showed you just a few minutes ago. And there's also a PDF file in here of the walkthrough for the analysis so that you can use that information. So all of this is contained here. Now, where am I? I'm in Cheese. Let me get out of Cheese. And let's take a look at this. Oh gosh, really? So what I have here for you is another data set called deodorant template. And this is actually a human microbiome experiment. And as you may imagine, it involves looking at deodorant samples. I was a little unsure about how this one would work when it was proposed by our students, but here we have it. I've put a README file in here for you so that you can understand what the sample was and how the experiments were done. And in this directory called task, there is nothing yet because I didn't put anything in there. I should probably have deleted that. But you will be able to, whoops, sorry. You can use this deodorant example and its associated information inside to take the worked example of cheese that we've given you, for which you have the recording and for which we've guided you through the analysis, and you can do deodorant from first principles for yourself, using all the steps that Art described for setting what your sequencing depth is, looking at all the other aspects, and it similarly has what I need to put into it is the task, the questions that we saw, like we saw them for cheese template. So this gives you an opportunity to not only be guided through something, but to do a new data set and conduct the analysis of it yourselves. So I hope that that has helped you understand how we can use high throughput technologies like next generation sequencing to generate data of sequence and genome information for bacteria that is medically relevant, increasingly medically relevant, and helps to highlight why we are starting to use computing and increasingly sophisticated computing for these analyses. I just want to check, has anybody managed to get their fourth script to complete and to run? I'm going to stop sharing my screen now. Has everybody got their fourth script sorted and run? Art, there's a question about not being able to find the file QZV.
Okay. That I just when I wrote that, I just meant a generic file name. So, for example, if you have a file called dmux.qzv, just type in dmux.qzv rather than file.qzv. That's just a generic name.
And maybe it might be worth, you were showing the visualizations in Chrome in a browser. Would it be possible just to quickly step through the output of the fourth one and show how to access that for people who won't have had a chance to download the walkthrough yet?
Yeah, sure. I can show you how to copy files over to your local system and then how to load them up, the visualizations.
Well, I think people should be okay with copying files to their local systems. Maybe you could just load up a visualization for us. Would that be okay?
Okay, cool. Yep.
Because that way everybody will have seen the, from start to finish, end to end.
Yes. 1 second. I just need to...
Sorry, I didn't mean to put Art under pressure. Shabnam, Art's just going to show you that in a second when he gets himself sorted out, because I've asked him to do something that he didn't anticipate, but he has the files and I don't, so I can't do this one myself. I actually probably do have them somewhere. Maybe I should have. Now you're going to find them faster.
Okay, cool. So wait, I'll just share my screen quickly. Right. Okay, so once you've got your files onto your local system, you just go onto this website called view.chime2.org.
And this is in the presentation.
Yes. And then when you're in view.chime2.org, you just go to the file that you want to visualize and you drag and drop it into the web interface. And it's that simple. It will just load the interactive visualization file and you can analyze the statistics from here.
Excellent.
Yep.
So does anybody have questions about any of what we've done here? Are we all good? We're coming to the end of this session. If everybody's comfortable that they have appreciated the guided walk through this session and feel confident that they could continue it on their own if they've not done it here now by reviewing the recording, and might be interested in carrying forward and doing the unknown data set that you've just been given, the deodorant one, could we have some hands up just to see that everybody's happy to proceed to the next step of the molecular medicine workshop?